The question is, can a basic science laboratory help you with your clinical trial project? The answer is yes. My name is Jose Antonio Diaz. I'm a vascular surgeon scientist leading a basic science laboratory in the United States. Please let me walk you over some collaboration opportunities that can be established between basic scientists and clinical trialists. The ideal scenario is a two-way collaboration where a basic scientist and a clinician can help each other in a collaborative effort to first deliver a good proposal and then conduct a great project. There are several aspects where basic scientists can contribute to clinical research, but I will mention two, probably the most important ones, during the research planning and during the research process. Let's talk about research planning. Several clinical trials results have been contradictory, not only because after the results were analyzed, conclusions reflected unexpected outcomes, but because failure in the experimental design were detected. Don't take me wrong, clinical trialists are trained in experimental design. However, there are areas in medicine where the input of a basic scientist could bring to light some aspects of the disease that sometimes practitioners are not totally aware of, and vice versa. At the end of the day, clinical trials can serve as an opportunity to use some resources from the basic science to test the hypotheses that basic science laboratory level are beyond to the level of the clinical laboratory. Those basic science laboratory actions may involve cell culture approach, for example, the use of preclinical models, the use of banked samples. They can further test the hypothesis with tools that are not always available in the clinical setting, which is the hospital. This type of research planning collaboration adds to the clinical trials an extra set of tools that not only make an application more attractive, but also open the opportunity to further explore the hypothesis to be tested. I particularly support the inclusion of basic science component in a clinical trial where such addition can play a critical role. Unfortunately, this is not commonly seen. So the take-home message of this first part, which is the question of whether a basic scientist can help in a clinical trial during research planning, is yes. Let's talk about during the research process. Clinicians and basic scientists that are currently working together in collaborative efforts better understand venous and lymphatic diseases. For instance, a basic science laboratory could contribute with a test of protein biomarkers. A biomarker is a measurable indicator of a specific biological state. Biomarkers can be used as a tool for diagnosis to detect the presence of the disease or follow-up to detect if a treatment is working on a patient. These biological biomarkers, can be measured in several tissues, including blood. There are several technologies or assays available to determine the levels of biomarkers in tissue, including, but not limited to, the omics for metabolites, metabolomics, or proteins, proteomics, the expression of genes, gene expression tests such as the quantitative real-time polymerase chain reaction or QRT-PCR, the enzyme-like immunosorbent assay or ELISA, preclinical models to study a biological process, a treatment, or a new drug. Please, let me walk you through some of these tools. Let's talk about ELISA and blood collection. The ELISA is a test that allows researchers to quantify the levels of biomarker in, let's say, plasma. If we are interested in measuring the levels of an inflammatory biomarker, we can use the clinical laboratory for some biomarker in the hospital, but there are some research-specific biomarkers that can be measured only in a basic science environment due to its current preclinical phase, for example. In other words, biomarkers that are not commonly used in the clinical setting. Let me give you an example. Let's say we would like to measure P-selectin, which is an addition molecule that participates in leukocyte rolling and attachment to the vein wall during deep vein thrombosis initiation. P-selectin is attached to the plasmatic membrane of the endothelial cells, and this location makes its determination in plasma very difficult. However, there is a soluble component of this molecule named soluble P-selectin that can be measured freely in plasma. Let's talk about blood collection and plasma processing. We first obtain blood from a vein and immediately place the blood in a tube pre-filled with an anticoagulant. This anticoagulant should not interfere with the determination process, and this is very important. For example, for soluble P-selectin, EDTA, which stands for ethylene diamine tetraacetic acid, is the anticoagulant that won't interfere with the biomarker. It is important to mention that some anticoagulants used to prevent blood coagulation after collection can interfere negatively to a detection method. Thus, if you use a plasma to determine a biomarker, make sure that the anticoagulant is appropriate. In other words, use the right blood anticoagulant. Anticoagulants commonly used in research are EDTA, sodium citrate, ECD, which stands for acid citrate dextrose, heparin, and they are easy to recognize by the color code in the cap. Purple cap for EDTA, light blue cap for sodium citrate, yellow cap for ACD, or green cap for sodium heparin. Once blood is collected, proper centrifugation to separate plasma is required. Then the sample is ready to be analyzed. So let's go back to ELISA, and I will walk you in the basics of what is called sandwich ELISA. We use a plate, and the plate that has wells on has multiple wells, 96, and each of them had been coated with an antibody that is called detector or primary antibody. The antigen, the molecule whose concentration in blood we are trying to determine, is present in plasma. So we add the plasma into the wells. The captured antibody selectively binds to the molecule we are interested in. Then we wash out the plasma, and the captured antibody will stay in the well with the molecule we are interested in, which is attached to. At this step, we cannot see the concentration. In fact, the plate looks transparent to your eye. In order to visualize the concentration, we need another step. We add a secondary antibody, which is carried an enzyme named peroxidase. This secondary antibody selectively gets attached to the molecule that remain attached to the primary antibody in the well. Then a substrate for the peroxidase is added. The reaction of the peroxidase with the substrate provides a color to the well. The darker the well, the more concentrated the molecule of interest is. A plate reader is a device that is used to detect chemical reactions by measuring absorbance. Using a plate reader, we can measure the concentration of the molecule we are interested in this case, soluble P-selectin. Finally, additional assays can be incorporated to a clinical trial to help testing your hypothesis. For example, the use of preclinical models to study a biological process, a treatment, or a new drug. These are very useful, not expensive in general, very reliable, and require a high level of expertise in preclinical settings. Preclinical models are well-recognized methods that have been helping to understand diseases and discover new treatments for centuries. For a screening of a new biomarker, metabolomics or proteomics could be useful for exploratory studies. This requires high level of expertise in the area, high level technology, and sometimes can be costly, but worth it. Other basic science laboratory assays are determination of signaling using gene expression, such as the Q real-time PCR, which is a powerful research tool for molecular biology laboratories with many different applications. At the cell level, the information is stored at the DNA. The gene can be expressed or repressed. This can be measured by number of RNA copies using methods such as the Q real-time PCR. So the take-home message of this second part, which is the question of whether a basic science can help in a clinical trial during the research process, is yes. Thank you very much. And remember, we get further working together.